This is a production of Cornell University.
Very generous presentation, very kind words. Thank you very much. And I'm so glad to return to Ithaca and to the plant pathology department. I had great time here. I met my husband in the department of plant pathology. My son was born here. So I have very good memories of my time in Ithaca. So they probably know my husband will join me here in Ithaca by the end of June. And so I'm really glad to come here and see so many friends around. So I'm going to talk today only about Phytophthora. So sorry for the bacteriologist, and I also work with viruses at some point of my career, but I'm only going to talk about Phytophthora today. And only in the case of Solanaceous and Phytophthora species, I have worked also in other crops with the interaction with Phytophthora in avocado and other crops, but I'm going to focus today in this interaction. So in my PhD, I work with bacterial disease, a cassava bacterial disease in South America and Africa, and developing some resistance to the CBV caused by Xanthomonas axonopolis, Phytophthora manifoldis. And then when I came as a postdoc to Bill's lab and then to Chris Mark's lab in Geneva, I worked with Phytophthora infestans. And we were studying the interactions between the host and the pathogen. And we were interested in the compatible interaction. A lot of labs were dealing with the resistance, trying to understand the resistance to Phytophthora, but we decided to focus on the compatible interaction, where the disease is happening. So we started doing some studies with potato, and we found a role for the carbonic anhydrase. And it was my first collaboration with betacei, with Greg Martin and people in his lab. And we found a role for this enzyme, the carbonic anhydrase, that when we silence this enzyme in plants, we saw that the pathogen, that Phytophthora infestans, grew more quickly, indicating that suppression of the carbonic anhydrase can increase the susceptibility to P. infestans. And that was interesting, but more interesting to me was that the largest group of genes suppressed during the compatible interaction were photosynthesis related. And we wanted to understand how or why we can imagine that the plant shuts down the photosynthesis, so the plant can dedicate all the efforts to the resistance, so to resist the attack. When I went to Colombia, we approached this question with a more broader focus, and we used this metabolic reconstruction as a novel approach for this characterization of these interactions. And we did a reconstruction of the metabolism of the plant during the compatible interaction. And this is very, very, I think it's amazing to see how the metabolism of the plant changes during the time course of the disease. Each spot you see here is a metabolite. So you can see how the metabolism changes drastically, and then it recovers, then it changes again, then it recovers, and then finally the plant will die. But the plant is trying to adjust the metabolism to resist to this attack. We found a connection with the photosynthesis through this pathway. So the good thing with these metabolic reconstructions is that you can explore each pathway if it's suppressed or activated during the interaction. And we found that probably one of the key to this photosynthesis suppression was lying in the glutamate synthesis reduction. We know that glutamate is involved in the chlorophyll synthesis and other steps, but we found a very good key there. Unfortunately, I couldn't continue this study because we didn't have funds for this study. But while I was here also in Cornell, we did something that was probably very, risky. We did a transcriptomics analysis in the field. I think that people probably thought at the moment that we were crazy to do a transcriptomics analysis with plants in the field. They said, you are going to have so much background of the expression, you will not see the differences in the response of the plant. And you can see that our field in Freeville had more flags than plants, because we had to identify each of the plants with the treatments, and we repeated this experiment 5 times over the season. And we could find some expression even with a big background. We saw a lot of expression coming only to put the plants in the field. But over that background, we could see that some genes, and I'm showing only 2 of them, that showed an expression in the resistant cultivar over the susceptible one. We silenced those genes, and we saw that silencing those genes, they reduce the resistance in the resistant cultivar. So from my studies here at Cornell, and with this study of these interactions, I can have 2 take-home messages. The first one is we have to try to integrate functional genomic information and biochemical knowledge, so we can see and understand the properties that arise from the interaction between the metabolic systems components and in response to the environment and the behavior over time. So I don't know if the metabolic reconstructions are the answers. They are models, and models have their ups and downs and their good things and bad things, but at least we can see a broader picture of the interaction with these metabolic reconstructions. For that study, we collaborate with people that have these metabolic reconstructions very well adapted to their systems in bacteria. It was the first time we were trying to do it in a more complex genome, like the one in Phytophthora. And the second one, and Bill continued these studies in this, is that you have to study your plant pathogen interaction as close to real crop conditions as possible. We know that the highly controlled environments, like the ones in the greenhouse, grow chambers, may render the plant highly responsive to non-treatment subtle environmental cues that will hide the true responses to the pathogen. And we showed with our field in Freeville, that field experiments, that the field-grown plants and use field-grown plants does not preclude the detection of important difference. So try to do your experiments as close as to real conditions are possible. And then in 2004, I moved to Colombia. And in Colombia, my main focus of research was Phytophthora sensulato. I call it sensulato because when I arrived, I tried I think I was working only one species, that was Phytophthora infestans, and then we end up with 2 species. And when you work in Colombia, and Rebecca knows this very well, there are complex environments. We have potatoes all year long, and when you have the potato field, you can have in the garden of the grower all the other hosts of the pathogen. So nice, because you have the reservoir of the inoculum going all year long. So you have Cape gooseberries, you can have Solanum quitoense, you can have peppers, you can have Solanum betaceum. So you have all the hosts of the pathogen to the potato fields. So not surprisingly, when you start your crop, the Phytophthora shows up immediately. So I tried to study the pathogen in Colombia, and the last study of Phytophthora infestans, when I arrived in 2004, the last study dated like 15 years old, so we had to see what was going on. So we studied the host. We did a continuous monitoring of the populations. I'm gonna show you some studies in the population studies, genomics of the new species. We studied the pathogenicity and virulence determinants, and I'm gonna end up my seminar showing you some alternative ways to control Phytophthora infestans. So first, we wanted to establish all the hosts in Colombia. We have this huge diversity of solanaceous crops in Colombia, so we wanted to show or to see which were the plants to host Phytophthora. And we could show that in these solanaceous crops. Of course, in potato, we have the yellow potato, that is Solanum fureja. Some breeders call them Solanum tuberosum subspecies fureja, that is the yellow small potato. And the tomato, tree tomato, and I'm going to talk a lot about the the tree tomato case. Lulo, that is Solanum quitoense. Cape gooseberry, that here you can hear all other names like goldenberry, I think, or I call it Cape gooseberry, Physalis peruviana, and one wild species of Solanum, that is Solanum viana. And we started our population study. But first, let me give you a quick guide to understand the population of Phytophthora. So usually, this is a clonal pathogen. Usually, well, it's P. infestans is heterotype, so it has to have the two mating types to go to sexual reproduction. But usually in South America, you only find one of them. In some countries, you find only the A1 mating type. In other countries, you find the A2. But you don't find, it's very rare to find the two of them in a country. So we study this pathogen as clonal lineages, and all members that a clonal lineage is all members descend from a single individual. So we can have US1, US2, US3, etc. Depends on the time US1 was the first described in the US, and so on in Ecuador, Colombia, etc. And a clonal lineage is defined by several markers: the allozyme genotype, the RFLP fingerprint, mitochondrial type, that can be 1A, 2A, 1C, and the mating type. So once you have an isolate, you have to run all the tests to see to which clonal lineages it belongs. So in my first study, when I arrived, we published it 4 years later to my arrival. We characterized the population in Colombia. So apparently everything was EC1, the one found in Ecuador. So it corresponded to the clonal lineage EC1 that replaced the US1 population previously described 15 years before. And all the strains were A1. And we found among more than, in all my life, I think I collected more than 1,000 Phytophthora strains. And we only found one that was the A2 mating type. And it was associated to Physalis peruviana. And I described the disease, I swear, I saw it, I published it. And after that, I couldn't infect Physalis again. We even came to Cornell to put it in the field with a strong pathogen pressure, never got infected again. So that is a mystery that still haunts me at night. That is the Phytophthora or the Physalis. And I couldn't see any other A2 strain in Colombia anymore. So this was with the traditional markers, the EC1. And then we use genes to describe the population, to see how many haplotypes per gene we had in the population. We use mitochondrial genes, we use nuclear genes, we use an avirulence gene, and we saw that it was pretty very clonal, only one haplotype most of the time. And in Venezuela, it was so nice because it was like white and black. Like you had one haplotype in Venezuela and the other one in Colombia. It was really nice. And we had some diversity, but most of them were very, very good. And we saw this in the south of Colombia, an explosion of diversity of haplotypes in the south of Colombia. And I'm going to come back to that story later. And then we use what everybody was using at the moment, and then in 2019, the microsatellites. And again, from only one clonal lineage, EC1, we saw this explosion of diversity. Each color represents a genotype with the microsatellites. And you can see in different locations of only one state of Colombia, the huge diversity that you have. So what we have one RFLP fingerprint with 76 MLGs. So we have to understand why we have this diversity, and it corresponds to different levels of virulence. We still have to understand. But it was nice because we could see, or we could answer the question, if the haplotypes were correlated with the region or the location, or with the genotype of potato. And we couldn't see any association or any relation. You can see the haplotypes that are in color, that they, or the haplotypes that are the circles they have different origins, and you can see them in different regions. The same with the genotypes. The genotype, we could isolate different genotypes of genotypes of the pathogen from one genotype of the potato, of the host. So it was, there was not a correlation between geography, neither geography nor the potato gene. But the good thing after all this work is that with Carlos Newster, a plant breeder, potato breeder, we could release last year 3 new varieties resistant to Phytophthora in Colombia. So all these years, all this monitoring, all this trying to identify the different individuals and variants help us to identify the resistance in these varieties of potato. And we also monitor the resistance or susceptibility to different fungicides in Colombia. So the most common fungicide is mefenoxam, and we saw a very high rate of resistance to this mefenoxam in Colombia. Very susceptibility to other compounds, but this was very amazing because in Colombia, they spray by calendar. They spray once or twice per week, regardless of the conditions, regardless of everything. And they don't know that we, or at least at that moment in 2019, we didn't know that we had this high level of resistance to this mefenoxam. We also studied the populations of tomato, the populations of Phytophthora infestans associated to tomato, and we found, and you see the ones in tomato with this dark blue, that are really separated from the ones from potato, that is in the light blue. And even you see in this network that they could even be in a way to speciation, probably a new species, we don't know, we didn't explore that, but they are really not connected, some of them, to the rest of the population of Phytophthora. We studied a question that was proposed very long time ago, that the strains isolated from tomato and potato have host preferences. And we indeed showed that the strains isolated from potato, when they are inoculated in potato, they cause more disease, higher level of disease. And when the isolates are isolated from tomato and they are inoculated in tomato, you see a very strong host preference to tomato than to potato. And when we saw what was the resistance of susceptibility of these tomato strains compared to the US strains to 3 different molecules, mefenoxam, simoxanil, and fluorocolid, we saw that we had, again, high levels of resistance in our strains to mefenoxam, and compared also to the US strains. In Colombia, we have high levels of resistance to mefenoxam. And doing those experiments, and this was the work of Juliana Gonzalez here present in the Fry Lab, they discovered they described also in the Fry Lab a phenomenon that also was described a long time ago, that is the mefenoxam acquired resistance. When you grow your isolates in different concentrations of the fungicide, you see that you can kill them, or they don't grow. But when you take inoculum from a super little concentration of mefenoxam, and you place it in a higher concentration, you see this very rapid acquired resistance to the fungus. And it occurred, and it was Juliana's paper during her undergraduate studies. It occurred in different Phytophthora species and genotypes, and seems to be unique to this mefenoxam. It didn't show with other molecules. So she did a more in-depth study, reanalyzing whole transcriptomic profiles of strains that were submitted to this mefenoxam in silico search for alternative POL1 subunit, RNA POL1 subunits in the genome of PI. Probably Phytophthora was using other subunits to resist to this fungicide. She also did some QRT PCR profiles, and she analyzed also the small non-coding RNAs to see if there was some epigenetics phenomenon going. And she came up with this nice model that first she saw that there is a pleiotropic drug resistance that triggers a primary cellular response that maintains the pleiotropic drug resistance, but there were also evidence of some epigenetic mechanisms of this phenomenon. So I invite you to read her paper. It's nicely written and it's a very nice model of the disease. And then let me go back to the story of these Phytophthora populations in the south of Colombia. So you remember that we have this explosion of diversity. So we wanted to explore what was going on with this population. At the time, there were these species described in South America and Central America. We had in Mexico Phytophthora ecomi, Phytophthora mirabilis. And in South America, we had this new described species that was called P. andina. So probably we were dealing with two species in the south of Colombia, Phytophthora infestans and Phytophthora andina, and we wanted to know a little bit more. So we went back to literature and saw that in 2000, Maria Eugenia Ordóñez, a colleague of mine in Ecuador, had published this paper that she was reporting a novel population of Phytophthora. She didn't say infestans, it was sensulatum, similar to P. infestans attacking wild Solanum species in A. Attacking this species, it was A2, the mating type, for the first time in Ecuador, with a new mitochondrial haplotype, 1C, and they were neither pathogenic on potatoes nor tomatoes. And then, in 2004, this paper showed that they discovered, additional to the A1A mitochondrial haplotypes, they discovered another lineage that was an EC2 lineage with A2 and 1C. So at the moment, we had two clonal lineages of P. andina, EC2, EC3. Well, they didn't call yet P. andina. They said they were novel populations, EC2, EC3, US1, with different mitochondrial haplotypes and different mating types. Okay? And then this big paper was published on the phylogeny of Phytophthora, and they call it Phytophthora andina, a putative new species. But they say, and this is very important, that ambiguous gene sequences pattern of nuclear DNA in P. andina could indicate a hybrid origin. And that is interesting, because something was going on the nuclear DNA of this group of species. And then my friend, Ricardo Oliva, published this paper describing the species, Phytophthora andina. And they said, we have this set of strains, one EC2 associated with wild species, and the EC3 associated with Solanum betacei, the tree tomato. And personally, I didn't like this paper. I talked to Ricardo several times, and we sent a letter to the editor saying that there were not evidence to define a new species. And why not? Because first, they couldn't, we saw probably two different species of P. andina, one to the EC3 and EC2. So we knew that there was something in this diversity that was not a hybrid species. We were probably talking about two different species. When we took the data from the genomes of the mitochondrial genomes, and we saw that they were really different, these two, 1C and 1A. Because in P. infestans, within the same species, you can have different mitochondrial haplotypes, but these were really different. And these were also clustering with different species. So apparently they were very divergent. And when we run some genes, we couldn't differentiate P. andina of P. infestans. So in this letter to the editor, what we were saying is that we didn't think there were enough evidence to say that P. andina was a single species. And in Colombia, a group in the south of Colombia showed that with AFLP, they could show this separation of the ones associated with Solanum betacei and the one with potato. So we were dealing with several species. So we took several, we tried to get several lines of evidence to show that we were dealing with different species, actually with 3 different species. The first thing we did is to grow these 3 species, what we believe they were the 3 species. So at the moment, we call P. andina the ones associated with the wild species of Solanum, P. infestans, and P. betacei is the one associated with the EC3 group associated to Solanum betacei. So this is the EC3 group, and this is the EC2 group. And you see that when you use 3 tomato agar, the P. betacei grows very well compared to the other ones, but it doesn't grow at all in potato. So that was our first line of evidence. We also did a lot of morphological descriptions, and you see that usually, well, P. infestans sporangia look like this with the papilla. And in the sporangia of P. betacei, that is all of these, you couldn't see, well, you see all spores, but you couldn't see sometimes the papilla. So you see some intermediate sporangia without papilla. So we also saw some difference in the morphological descriptions. Then we did some phylogenetic studies, and we could see that P. andina and P. betacei were separated. And so these are in red the strains we collected, and in green we have the P. andina from Solanum betacei that was described in the Ricardo's paper. And we saw that they were separated with different tools. We also measured the content of DNA in the 3 species, and we see that in P. betacei we have the double content of DNA than in the other species. So you can see here the summary of the content in the genome. So we have more DNA in the nuclei of P. betacei than in the other species. And then in population studies, again, when you take the K and that shows the best fit, you can see the 3 populations separating, and here with these PAC, DAPC studies, you see really that they will separate the 3 species separate, with the P. andina associated with EC3 clustering with the ones isolated from 3 tomato in Colombia. And of course, we did all the infection assays. We inoculated everybody in every genotype, so the 3 species in we didn't have the wild Solanum plants in Colombia, so we couldn't inoculate, but we inoculated in tomato, potato, and tree tomato. And we showed again that P. betacei only can infect Solanum betacei, but cannot infect the other host. And also that P. infestant doesn't go very well in tree tomato. And the final line of evidence, like as a biologist, is to show that there is reproductive isolation. So we did all the crosses. This is hard work. Bill knows that, to do all these crosses and see if we had the formation of oospores of hybrid viability, if we have pre-mating isolation or post-zygotic isolation. And I'm gonna show you only the results of the P. infestans P. betacei, showing that the hybrid are absent. Some oospores are formed, but they are not viable. So the hybrids are absent, and we have the reproductive isolation. So with all these lines of evidence, we could publish that we have a new species within the clade 1C associated to Solanum betacei in Colombia. So P. andina in reality is 2 different, was 2 different species. P. andina corresponding to the EC2 with different mitochondrial genomes, and P. betacei, the one with EC3 associated with Solanum betacei. And we were in phase of an ecological speciations. The 3 species are reciprocally monophyletic, that is the final proof to have a new species, and the most probable scenario is a speciation with a secondary content. And we, after that, we saw that it is now clear that we needed a unified conceptual framework to properly define the species in plant pathology and characterize the divergent processes in these plant pathogens. So we try to to show this in this paper in the annual review of phytopathology, that studying the processes involving speciation, it boils down to understanding the barriers to the genetic exchange. We have to show the reproductive isolation. Most of the time it's pre-mating because there is one of them cannot infect or both of them cannot infect each other's host. So we have this pre-mating isolation because they are really isolated by the host. But sometimes you can have post-zygotic isolation when you don't have the viable or spores of viable hybrids showing. So we need to connect molecular taxonomy with reproductive isolation to properly define a species. Sometimes we only show phylogenetic species without showing if there's a barrier to this exchange. And the pathogen host range appears to be constrained by the pathogen effector repertoire, but we haven't studied the relation between the plant immunity genes and shaping the evolution of new plant pathogen species. And most of the times, the study of ecological and geographical drivers will lead to better understand of these events that cause the reproductive isolation. After that, we came back to the field to see if this new species was only in the south of Colombia or was in other parts. And we saw that it's also is in sympatry with Phytophthora infestans in the central part of Colombia. And again, with more strains, we again could demonstrate that we have the three different populations, P. infestans associated with different Solanum. We have the P. infestans, the tomato strains that we have probably to study them a little bit more because they separate from others, P. betacei and P. andina. So we have the different species. And probably you remember that we have twice the content of DNA in the nuclei. And so we went and sequenced the genome of this new species and showed that most of the genome was a whole genome duplication. The genome of P. infestans is huge, but it's mostly due to the repetitions and to all the transposons you find in the genome. But in P. betacei, you find a whole genome duplication. A lot of the contigs of the Phytophthora infestans are duplicated in the genome of the P. betacei, And we studied all the effectors. We have different kind of effectors, the apoplastic effectors, the crinkle ones, and the RX, RL. And we found that some of them are in both species, but some of them are unique to betacei. So now we have to show if these unique ones are the ones that allow betacei to thrive in Solanum betaceum. So we have in the three categories, some of them that are unique to betacei. Some are shared, and some are also unique to P. infestans. But most of them, and we found an explosion of effectors in this genome of P. betacei. So in the final minutes, I'm going to talk about also I collaborate with some biochemists to find novel solutions to control the disease or control Phytophthora. So we focus in this pathway, the de novo pyrimidine biosynthesis in Phytophthora. There are two ways that Phytophthora gets the pyrimidine with a de novo synthesis, but they also have the salvage pathway. So our hypothesis is that Phytophthora uses the de novo synthesis during the biotrophic stages of the disease. And then when it goes necrotrophic, it will go to the salvage pathway. We have to demonstrate. And why focusing on this pathway? Some of these enzymes have been attractive for drug development in humans and in Plasmodium. For example, this compound has been used to inhibit the fourth enzyme of the de novo synthesis pathway in some treatments to autoimmune diseases in humans. But also other compounds have been used to control the Plasmodium falciparum, the malaria parasite. And you know that Phytophthora is very close phylogenetically to this malaria pathogen, to Plasmodium. Is closer to this than to fungi. Everybody knows that, but is very close to the malaria pathogen. So, to answer the first question, we try to see the expression of the enzymes during the time course of the disease, and we could show that the de novo enzymes are all activated during the biotrophic part of the disease, and the salvage ones are downregulated. And then the salvage ones is the contrary. During the salvage pathway, the de novo synthesis enzymes are downregulated. Now, I'm starting again. So the de novo enzymes are activated. The de novo enzymes are activated during the biotrophic, and then downregulated during the salvage. The contrary to the during the necrotrophic part of the disease, that the de novo are downregulated and the salvage are upregulated. Yeah, very good. So we took one of these compounds I was talking about, the one that was used against the human enzyme. And we first showed that is localized, the enzyme is localized in the mitochondria. And then we tried to inhibit the growth of bee infestans with this compound. And we compare with the Crusade, that is a fungicide. And you can see that we could, increasing the dose of the compound, we could inhibit the growth of bee infestans, comparing very comparable to the fungicide. And something very important is we want to show that the enzyme from the potato and the enzyme from the pathogen had different sensitivities to the compound. And indeed, you see the scale, we show that they have different sensitivity. Is good because you will not inhibit the enzyme of the pathogen. You will only inhibit the enzyme of the pathogen. You will not inhibit the enzyme of the host, only the one in the pathogen. So, my final take-home message. In Colombia, agriculture is a very important sector of the economy, accounting for more than 16% of employment and 7.5% of the gross product. With significant challenges, low productivity, no data-driven, low adoption rates of technology, no impartial extension services, and only a very few of the gross domestic product is invested in research in Colombia. So, I highlighted the importance of continuously monitoring the pathogens population. With the funding we had, you see that we could only monitor the populations in 2000, in 2004, 2012, 2019. We couldn't do this continuous monitoring of the pathogen population. And we have to do that with the appropriate markers that allow discriminating variants at the local and global scale. We did that also with the Santomonas, and it's really nice how you can adjust the level of discrimination, adjusting the use of the markers that are available. Also, the importance of working with breeders to select the pathogens variants. We have, over my life, I work with breeders in cassava and potato, and we also end up with releasing new resistant varieties. And we need to understand the relationship between the genetic structure of the pathogens and the distribution of pathogenicity and virulence. But during my life, I also work with fungi. So I want to show you some pictures of my research with cordyceps, with amanitas, and with morels. And we found this in the bricks of the university. Really interesting. And this is really interesting because we could grow some in vitro. We have the secret. And then we also studied the microbiological diversity in endophytes. And I have sampled palms. I have sampled plants in very extreme environments in the Paramus. So, and this is always thank you to my collaborators around the world. Thank you very much.
Very nice.
Thank you.
Is the betacei genome an alto or allopolyploid?
I don't know, but we can collaborate and see. Yeah, it seems to be alloploid. When I also published a paper before I knew there was a new a species. We published this paper with Nick Grunwald in Oregon State, showing the high levels of heterozygosity of the genome. So I think it's an alloploid. Rebecca.
Thank you so much for that wonderful story. And I also take my hat off to you for being able to continue throughout while managing universities. So I was really, I mean, so much of that is fascinating, but the fact that your betacei tree, your
tree tomato
can grow on tree tomato agar, but really cannot grow on potato dextrose agar. Like, it seems sort of like either there's something that it needs that's not there, or there's something that it doesn't like that is there. So like, what is there sort of a straightforward nutritional problem that contributes to the host isolation?
I think it has to be with nutrition, not only with the effector repertoires, but also with nutrition. We haven't been able to isolate betacei from potatoes in the field.
If you put a little of that potato into the tree tomato agar, does it crush its ability?
I'm not gonna try. Yeah, probably something in the potato that found in the potato that yeah, but you cannot inoculate, like it doesn't go in leaves or yeah. So it has to be. I think it's not only the effector repertoire, as I say. I think there has to be something else. Yeah,
fancier explanation that seems necessary.
Yeah.
Bill. Hi. Oh, what's the range of tree tomato? And is Phytophthora betacei throughout the range of tree tomato?
Well, yes, but I can say that in the metric system. I'm sorry, I will. Yeah, tree tomato can go to very low, well, not lowlands. I would say from 1,000 to 2,000 meters above the sea level, and we could find all Phytophthora all over the range. Yes, because when I did this sampling, I started in the Magdalena River, very like 0 meters above the sea level, and went up to Bogota, that is 2,600, and we could find isolates all over I was doing another project in Collectotrichum, but I was sampling also Phytophthora, and we could see the Phytophthora betacei all over the range.
How about throughout South America, north to south?
We haven't done that, but then a paper was published in Peru showing different populations in using the phylogenetic tree, some branches, and I think one of these branches, they don't call it P. betacei, but I think one of the clusters is P. betacei. Yeah. So I think we discussed that with my friends in C. We were going to do that, but then BTI hired me, so I couldn't. Yes, Derek. No.
Yeah, go ahead. I was trying to see if there's anything.
I'm interested in the diversity in P. infestans that you see in Colombia. Do you attribute that to just the fact that it can persist year round and all those hosts are there, so it just multiplies a lot? Or do you think there's some other factor influencing that diversity?
But I think it's a plant biodiversity that we have, and all these Solanum species. We haven't, well, you know that most of Colombia is not explored because of the problems in security we had in them. But every time I go to the field, I describe a new species of fungi of Amanita, and I'm sure that in the plant side, there should be also an explosion of Solanum species. Like when Mexico, and Bill described this very well, in Mexico, you also have a lot of Solanum species, and they could describe different Phytophthora species as well. So it goes with the host. Adam?
I'm wondering if enough is known about sexual compatibility across Phytophthora. I think whether you might see after speciation, gene acquisition, mutation, hormonal retransfer, or whatever, that would reestablish sexual compatibility, right? And you know, allow for merging. It could be really relevant if we're thinking about potential for divergent sets of effectors that come together as one species. Divergent host specificity. Any insight you might have?
No, but it's something that is probably very doable right now. The problem with I don't think there is a good P&D genome right now. That is, we couldn't do a lot of comparisons with P&D because the genome that is available is not in good shape. We couldn't sequence again. But then I think it's a good thing to see the coalescence and all the divergences of the effector repertoires. Yes, we can do that study right now with the genome treatment. Yeah.
Other questions? No, I don't see any on the online. Okay, then let's thank Sylvia again, and may I remind the students and postdocs there's lunch with Sylvia in somewhere.
Somewhere.